Good morning, everybody. I'm here to present Professor Jerome de Groot. He is a senior lecturer at the School of Arts, Languages and Cultures of the University of Manchester in England. Jerome works on the representation of history in contemporary popular film, television, drama, and games. Also, he has works on the literature of the period of 1640 to 1660. His primary research interest is in contemporary popular history. Consuming History is a book published in 2008 and with a second edition in 2016, and Remaking History from 2015 are both published by Routledge and concern the ways in which contemporary popular culture engages with history. There are chapters on historical film, television, both documentary and drama, museums, computer games, reenactment, and novel writing. So, Professor.
Hello. Hello. Bom dia. I feel like I'm a rock star with this. And I'm wearing really cool trainers, so make sure you notice these. That's the most important thing. Thank you everyone for coming, and thank you particularly to the INTH and the SBTHH and everyone involved, Berber and Marcelo and Andre for introducing me. Is that better? Sorry. Yeah? Andre for introducing me, particularly to Guilherme Bianchi because he's been incredibly helpful to me and has responded to all of my very particular whims. And I think this is the last keynote. So I think as a group, we should thank the organising committee for their work and the time they put into this. When Berber asked me to speak at this conference, I started becoming quite scared, and increasingly I've been scared coming up to it, and then I've sat through papers and I've become even more scared. I'm not an historian, I'm not a theorist of history, I'm not a philosopher of history. I mean, I've been in rooms with some very smart people talking about some very, very difficult things that I've really appreciated, but it's not really what I do. So, the way I see it is I was invited because my work does is kind of link theory and practice. And what I hope I'm going to do today is to sort of round us up a little bit with a kind of consideration of those two things. My title is Double Helix History, and I'm speaking particularly about DNA and history. So, in his 2002 book, French DNA, Paul Rabinot argued, with the coming of biotechnology, not only new subjectivities and new apparatuses, but also new life forms, cross divides 19th century social thinkers convinced themselves were unbridgeable. Similarly, Paul Gilroy has suggested a new humanism as a consequence of biotechnological advance. At the smaller, the microscopic scales that open up the human body for scrutiny today, he says, race becomes something less meaningful, compelling, or salient to the basic task of healing and protecting ourselves. And I want to take up these points and suggest that biotechnology, and particularly improvements in DNA sequencing, prompt new ways of thinking about the human and their relationship to the past. Now, to do this, I'm going to think about what Jackie Pearson and others have termed the genetic imaginary. That is, popular understanding of the work of our DNA, and more particularly, how we understand, represent, and visualize DNA. In particular, I'm interested in how genetic science interacts with historical awareness. Therefore, this paper investigates the intersection of genetics and popular narratives of the self and the past. What I'm going to do is look at various manifestations of DNA in historical investigation and think about how this relates to genealogy, humanness, public history, and the contemporary historical imagination. And so this is my structure here. I speak first about ancient DNA and the sequencing of DNA taken from human remains at archaeological sites. The second section looks at leisure DNA, that is DNA sequencing for genealogy, for family history. And my third section, my final section, seeks to sort of locate some of these ideas in wider discussions within contemporary global humanities. So I'm more generally interested in the materiality of DNA. How is it conceptualized and rendered and imagined? How is it both past and current? Something which veers between something reified as ancient and at the same time almost excessively modern. The duality of DNA in the historical imagination, evidence that lives within us, allows a dynamic connection with the past. Christine Kennelly, this is a popular science book, writes this: DNA in our life experience makes our bodies palimpsests. As we learn how to interpret the body in the context of its genetic code, we begin to understand how the hand of fate, the choice of families, and the enormous journey of DNA through deep time affect our lives right now. And you can already see the kind of rhetoric that's being used around it here. So many scholars have written, about the new identity politics raised by DNA science. And it's important to see these new identities, such as they are, within temporality. DNA brings genetics into a particularly historicized knowability. What was remote is now intimate. What was once science is now life. Kennelly writes, it explains our immediate family in the context of the human family and vice versa. And this sense of identity being related to temporality, that is the wider time of the human, and taxonomy, the genus Homo sapiens, allows a keen sense that we can move from understanding ancient humans to the now. Our genetic makeup allows an enormous historical perspective to open up. Yet the tools of analysis and conceptualization that are deployed in investigating DNA are little understood. I challenge anyone here to really know what goes on in these things. No, I don't. The narrative that is constructed here depends on a diversity of analysis, on probability, on mathematical modeling. The results are presented in a variety of formats and interpreted by scientists rather more than by humanities scholars. The evidence is clear, it is us, but it's also unreadable and unknowable. And the question of our individual relation to humanness is provoked by DNA investigation. How does race, kinship, ethnicity, identity function in this context? How does this change our understanding of ourselves in time? So my first section starts with an example. The most famous Anglophone moment when genetic science and history recently came together was the discovery, exhumation, and examination of King Richard III's remains in 2012. King Richard III, a English king, who obviously was written about by Shakespeare. They found his body in a car park in Leicester, the glamour, and identified the body through laborious DNA investigation. And this prompted a set of discussions in the press and beyond about the nature of lineage, the contemporary world's relationship with heritage, and the ethics and legality of disinterring human remains. The body of the king, as you can see here, was reinterred in Leicester Cathedral, but only after a year of legal wrangling about the final burial site and a final judicial review made by the High Court of the country. They were fighting over his body, literally. The 5 days of events celebrating this reinterment situated the burial at the intersection of heritage performance, nationalistic memorial service, and historical reenactment. The procession toward the final rest was attended by thousands of onlookers, and the coffin went through the town of Leicester with, as you can see, thousands of people watching. At the final service, Benedict Cumberbatch, better known as Sherlock, read a poem written by the UK poet laureate Carol Ann Duffy. Carol Ann Duffy's public poem, a sonnet, explicitly references Richard's body being brought from the mud. My bones scripted in light upon cold soil, a human braille. Andre's going to say this word for me in Portuguese.
Braille.
Everyone okay with what this is? But it's very important to me, the idea, because this idea of a kind of a text which is not a text, which is somehow tactile, somehow to be felt and also understood. I think Braille is a brilliant word for her to use here. And the sense of the interrelationship between language, skeleton, writing, mud, Braille, figures the complexity of Richard now as a thing and a dream, something imaginary but real. So the investigation into Richard III's remains was a complex project intellectually, scientifically, and ethically. The work on the DNA of the skeleton was crucial in establishing the veracity of the bones. And this event highlighted an increasingly important aspect of the configuration of memory and history in the cultural imagination. That is the decisive contribution of genetic science to establishing fact. It is the case that for decades, historians and various types of scientists have interacted to investigate, for instance, disease, environment and weather, emotion, evolutionary psychology, bioarchaeology, and more recently, network analysis and data mining. Many historians have thought quite carefully about how to engage with and develop new techniques and approaches. Yet DNA science, I argue, has become far more embedded in historical investigation than these other types of interdisciplinary approach. And this is mostly driven by the geneticists themselves. Joseph Pickrell and David Reich, two leading lights in historical genetics, argued in 2014 that improved technology and analysis technique leads directly to new ways of understanding human history, as they say here. Because of these technological advances, the past few years have seen a dramatic increase in the quantity of data available for learning about human history. Equally important has been rapid innovation in methods for making inference from these data. We argue here in their article that the technological breakthroughs of the past few years motivate a systematic revaluation of human history using modern genomic tools, a new history and geography of human genes that exploits many orders of magnitude more data than the original synthesis. This is quite a claim they're making here. So the DNA that they investigate and the modes of interrogation, the upgrade in technique, the opening up of quantity, leads to a richer understanding of how the species develops over time. And as you can see, the rhetoric is about accuracy, density of data, innovation, and the way that this allows a more textured understanding of human history. Now, much of this is driven by improvement in investigative technique. The revolution of new next-generation sequencing means that acquiring the entire genome is much cheaper than formerly. And it has a secondary effect of being particularly useful for sequencing old and degraded DNA, such as is found in ancient sources. Increasing computing power as well has also made the process much quicker, much more efficient, has given it increased resolution. Unlike Richard III, the remains of the boy known as Anzic I were reburied with much less fuss. The child's remains were discovered on a farm in Montana in 1968, buried with many artifacts identified as part of the Clovis culture. Named after the family who owned the farm, the remains, which had been buried around 12,000 years ago, were sequenced for DNA in 2014 before being reinterred. And the investigation suggested that Anzic I was both a direct ancestor of contemporary Native Americans and also had Asian ancestors, or as we would call them, Asian ancestors. And this work decisively challenges the Solutrean hypothesis that Native Americans moved to the continent of North America from Europe. It's much more complicated than that, but... So the sequencing of Anzic I's DNA was the final process of investigating the remains before they were reinterred. The ceremony was brief and low-key, but still provoked much debate about the use and excavation of human remains. Our man Minthorn of the local Umatilla tribe said, "These are our ancestors' remains, they are not artifacts." Quite a kind of precise understanding of what's going on here, and Native Americans are very kind of intellectually tooled up about this stuff, rightly so. I hope that the people who come after us remember this as well. So the investigation of ancient DNA like this is controversial for various reasons. The investigative team in this case, based at the Centre for Geogenetics at the University of Copenhagen, have worked on a variety of projects, including ancient plant DNA and the controversial Kennewick man. Their director, Esker Willerslev, there he is, great picture, was recently profiled in the New York Times, which argued, "The findings have enriched our understanding of prehistory, shedding light on human development with evidence that can't be found in pottery shards or studies of living cultures." And this sense that DNA can contribute more to our understanding of what is at present a relatively unknown past through the interpretative clarity of science is common when reporting genetic investigation. Although this sentence kind of echoes Pickrell and Reich, really, in terms of what they're saying. Now, one of Willeslev's early high-profile successes was the sequencing of the Inuk or Sakak genome from hair dug out of the permafrost in Kirketassusuk, Greenland in 1986. Yes, I did practice how to say that. The genome was so complete that the team could suggest hair color, baldness, and features. And this visualization, which was quite circulated around the world, was key in communication of the image of the 4,000-year-old DNA, even on the cover of Nature. Such analysis allows us to see human data see a human, as data and an image, as something idealized and something specific. This is what it actually looks like in the report, the DNA report. It also demonstrates, so you can see, quite different. It also demonstrates the power of the image over the data and the trust that is being invested in the scientific community. Much of the work that's done by Willeslev's lab is way beyond the layperson to understand. It's highly specialized, it's technical. The conclusions are worked through in exacting detail. This is amazingly profound science work. But the knock-on effect through media coverage and the circulation of things like the Sakak image are much less subtle. This analysis allows for the complication of the narrative of the development of Homo sapien. But there's even more complication when other parts of the human family are analysed. Work on 430,000-year-old bone fragments from the Sima hominins, the oldest DNA sample ever analysed, suggested that these early Homo neanderthalensis were separated from Homo sapien earlier than was thought. So they're Neanderthals and their genus is separated from Homo sapien earlier than was thought. And hence they were living together at the same time for longer. Basically, two types of humans living together for longer. Evidence is being presented here that is shifting the ways that we might think about the evolution of Homo sapien, about when the human, as we might recognize it, comes into being. If then we need to think about ourselves as a species or the human as a species, then there's a lack of understanding about when that species became defined, how old it is, and how its own ancestry actually works. So considering the use of DNA in these historical and archaeological investigations takes us back to key issues in historiography: evidence, analysis, narrative, materiality, interpretation. The work that's being done in Copenhagen and elsewhere on ancient animal and plant DNA seeks to investigate as accurately as possible using contemporary technique. The work includes high-level DNA analysis, statistical investigation, mathematical modeling, consideration of carbon dating, population genetics, cranial morphology, and many, many other types of technique. So these numerous techniques are brought to bear on the original DNA sample, this bone fragment. But the purpose of the work is to exemplify it, make it bigger and give it meaning. This allows the sample to live as both ancient and contemporary signifier. It's both old and in the now, interpreted and inert. The lab painstakingly reveals, through investigation and analysis, new possibilities about the human. And we have, I think, to see the work that is being done here as historical investigation of a kind, and to therefore theorize it. To demonstrate this, we might briefly consider the use of the phrases DNA archive or ancient DNA libraries in much of the literature. A DNA library is something precise and technical, involving fragments of DNA stored in microorganisms. To test and sequence DNA, such libraries are built from individual samples. So the library is in fact a set of replicated, propagated DNA strands. The library is actually a multiple copying of the original. The library terminology refers to the process of sequencing, shows us that the evidence base, or the data set, is the original sample which has been replicated. It's then made into data, or interpreted as data. In the example of the Sakak, for example, the original sample was a strand of hair preserved in the permafrost. In the case of Kenwick man, the original sample was 200 milligrams of a metacarpal bone, and this is the Sima skeleton's bone as well. Here, the material, that is, the original piece of the human, hair or bone, is reduced to its genetic detail and then replicated in order to be tested. And this presents us with a number of questions. How real is this evidence? DNA replication for this purpose is standard, but it means that the original and the created are in a complex place. What is the evidence here? What is the text to be considered? There's something material to look at and to fit into a network through consideration of comparative genetic analysis, algorithmic modeling, statistical comparison. Yet the material being sequenced, being imagined, being tested, being conceptualized, it's not necessarily the original thing itself, but something that's an identical version, paradoxically. How is DNA evidence then evidence in historical terms? What's the status of this information? How is the information that's extrapolated from it understood, imagined, and represented? So the genetic scientist is increasingly intervening into a set of debates that seek to understand humanness and to present the investigation of the past as the acquisition, organization, and interpretation of particular data. The sequencing of remains that are over 10,000 years old, that is from before the estimated period of the last glacial maximum, involve an engagement with controversy and argument about evolution, prehistory, humanness, and the definition of species. The development of enhanced genetic tools for analyzing ancient materials has led to an acceleration of information regarding human evolution and development. And this has a concomitant impact on the way that culture thinks about and visualizes humanness and prehistory. So genetic science, therefore, is establishing a sense of historicity, linear, structural, networked, factual, that interacts with evolutionary science, molecular biology, statistical analysis, and data mining. It's contributing to a definition of time, augmenting our sense of population development, and at its most baroque, arguing for a new way of thinking about humanness and history. Excuse me. That was the hard science bit. We move on a little bit. Where am I? Right. Yeah. So, this is me. This is my DNA. You may not notice it from our being up here. So, whilst ancient DNA testing allows us to think about genetics, section 2. And history, it's hardly a popular pursuit. Yet thousands of people are having their DNA sequenced for leisure purposes, mainly investigating their family history, as I said. So this phenomenon allows us to further complicate our sense of how double helix history is being communicated and understood around the world. What I'm concerned with for this section of the paper is the resonance of this kind of inquiry in the popular imagination. Genealogy is one of the biggest and most profitable activities on the planet, generally undertaken via massive gateway websites like Ancestry.com, which claims 14 billion family history records. It involves investigators around the world formulating a family tree and imagining their relationship to the past. Increasingly, these websites are adding a new tool to the researcher's armory, DNA sequencing. The armchair genealogist investigates their past by spitting into a tube. The creation of huge repositories of DNA information about database users allows for data analysis to be undertaken, leading to speculation regarding the ancestry of the individual. And of course, visualization of this. I don't get anything like the other stuff. This is what you get if you do this sequence. So it's already interpreted it for you. Famously, eminent African-American historian Henry Louis Gates Jr. articulates this sense of excitement for the family historian, but also the keen problems inherent in the archive. Searching for my ancestry was always a fraught process, he writes. Always a mix of joy, frustration, and outrage, as the reconstruction of their history, individually and collectively, must always be for any African-American. In place of the archive, he publicly places faith in science. His TV series, In Search of Our Roots and Finding Your Roots, used individual DNA sequencing to enable participants to look beyond the state collection of data. He argued that, and this is pretty extreme, and I think right, for the first time since the 17th century, we are able, symbolically at least, to reverse the Middle Passage. And this has historiographical consequence. Restoring the stories of the lives of the members of our extended families can directly transform the way that historians reassemble the larger narrative of the history of our people. What is at stake here is participation in history, the reinsertion of an entire set of people who have been intentionally effaced. So DNA sequencing is the tool of genealogical investigation. It's part of a suite of ways of imagining a physical, material relationship to the past, allowing the user to access a very different sense of their family history. Yet what Ancestry.com and other websites seek is the creation of a live social network based upon DNA sequences. So this is what happened when I had my DNA sequenced. Someone got in touch with me and said, I think we're related. And we are. Whatever that means. Through social media software, types might be matched. Ancestry.com seeks to enable the ordinary person to participate in this reclamation of identity. The archive increases through each user placing their sequence within it. The software can then match users, suggesting relationships that are previously unknown. And this seems an unprecedented medicalization of social relationships online. But I can talk about that in questions if you like. Furthermore, I think something what happens when DNA is realized digitally. DNA becomes an artifact, something material, but never to be touched or seen. It's just turned into numbers, figures, projections. Something rendered within the the network of connections, databases, social media, interrelationships. The already complex material between the material and the ontological becomes augmented and visualized through this digital software. Modern genealogy work materializes DNA in the digital through the rendering of results, and the physical, the experience of the individual. That's me. How this kind of interrelates, I find quite fascinating. So the unknowable becomes recognizable, and we see ourselves historically and immediately. The use of genealogical information online by big providers is part of a retrospective privatization of public historical space and the space of the body, and I've argued this elsewhere. Historical information is made to work for profit. If you do this with Ancestry, this is what you sign up for. Perpetual royalty-free worldwide transferable license to use your DNA in any form or context we deem appropriate, with any technology or device is now known or hereafter developed or discovered. It's kind of the future is already acclaiming your body. Amazing. Not only is the user's work being commodified, so too is their genetic code and that of their ancestors. The dead are resurrected through DNA sequencing to mean again, but also to earn again. Similarly, the body of the contemporary user becomes valued. Their DNA is valued because of the work it undertakes in contributing to this archive, which gets bigger and is self-generating. The database imagines the user archivally and physically, seeking to reveal relationships hitherto undisclosed, rerouting the individual in the contemporary, through the past, and then again in the now. The connection between people becomes a network event constructed by the algorithm, matching two possible users. So as DNA evidence becomes more commonly used, I think we'll experience more regularly the human as data, something expressed through numbers, and data that becomes something that's shared online. And this is a smaller section of a much wider phenomenon that's happening around the world. The interrelationship between the user of the service, their data, their network profile, and their actual body is highly complex. As a side issue here, new cutting-edge genetics is seeking to store information using DNA. Artificially created DNA stores information using the 5 DNA letters and is capable of keeping information for millennia, given the right storage conditions. This is Shakespeare, written in DNA. Sorry, where am I? It opens up possibility for writing human knowledge. Robert Grass, who's one of the pioneers of this, argues, if you look at how we look at the Middle Ages, it's very influenced by what information is stored. It's very important that we get a relatively neutral documentation of our current time and store that. Yeah. The point here is that the scientist historian is becoming the scientist archivist, developing new ways of storing and reading human knowledge. DNA allows a reimagination of the past, the present, and now the future. So my interest, to the concluding section, my interest in the relation between the material and the object, the concern with the multiple aspects of the thing when looking at DNA, is part of a wider discussion undertaken by scholars in the humanities about matter, imagination, and science. Jonathan Gill Harris's influential reading of Michael Serres focuses on the polychronic and multitemporal nature of the object. The point is the ethical and political duty to provoke a new discussion in the humanities through a consideration of multiple contending temporalities. Gill Harris derives his argument from philosophy of science to make the case that an object is never of a singular moment, but instead combines ingredients from several times. And we've kind of heard about quite a lot of this over the last couple of days. His point, built in this case from Bruno Latour's idea of the polytemporal, is that in considering the multiple times of the object, the scholar can begin to evade linear norms and directional templates of thinking. As Latour argues, we do have a future and a past, but the future takes the form of a circle expanding in all directions, and the past is not surpassed, but revisited, repeated, surrounded, protected, recombined, reinterpreted, and reshuffled. This sense of the interrelationship between the thing or event and the multi-temporal relationships between them and the ways they might be arranged allows a conceptualization of the material and its status in time. And to illustrate this, Latour famously turns to genetics. Some of my genes, he says here at the bottom, are 500 million years old, others 3 million, others 100,000 years. And my habits range from a few days to several thousand years. It's this exchange that defines us. In this light, we might begin, as Latour does, to consider DNA as something that is multi-temporal, and our analysis and understanding of it to be aware of this shifting. DNA is something that proves, following Latour, that we have never been modern. Indeed, the concept of modernity is problematic and possibly unethical, according to him. It does not allow us to consider our actual polytemporal natures. And we might also look at the little quoted section before this. Here we go. Let us suppose, for example, that we're going to regroup the contemporary events along a spiral rather than a line. Elements that appear remote if we follow the spiral may turn out to be quite nearby if we compare loops. The challenge to linearity that Latour outlines here is a line that lives in two plane dimensions and which bears some resemblance to a helix, helix oftentimes translated as spiral. In this telling, the event is a flexible thing, flickering in and out of temporal actualization. And in his later work, Latour discusses idealized materialism in his attack on the way that the whole machine has been conceptualized. He points out the relationship between the idealized image and the material thing itself, and that the interrelationship between the two states highlights the problem of iteration. Latour's work, as shown by Gill Harris's reading, is part of a shift toward what many have termed the new materialism, drawing from actor network theory, posthumanism, feminism, and postcolonialism. And in particular, new materialism stages the engagement between contemporary science and the new humanities. Karen Barad, one of the key proponents of this materialist movement, argues, I am seeking an epistemological, ontological, ethical framework that provides an understanding of the role of the human and non-human, material and discursive, and natural and cultural factors in scientific and other social material practices, thereby moving such considerations beyond the well-worn debates that pit constructivism against realism, agency against structure, idealism against materialism. So the challenge for us is to look at the way that the material and the imaginative are combined in relation to DNA. How do we account for the very real problem of the materiality of DNA, both physically material and imaginative? What is the legibility? How does it sit in time? Is it something polytemporal, as we might suggest from these readings? How can we think about it materially and theoretically? How about the data that are presented to us as evidence of DNA? Can we read these non-human but very human figures? Can we aestheticize them outside of scientific discourse? What's the political dimension of this data? Can we read it? The two examples I have looked at allow two distinct conceptualizations of the past. One highly personalized, one much more abstract. The human that emerges from these two conceptualizations is both concrete and imagined, conceptualized and constructed. Understanding them both as somehow simultaneous entails positing the epistemological ontological framework that Barad seeks. The further question, though, is how this impacts upon the way that those engaged with DNA investigation conceptualize their past, their relationship to it, and the future. So, as I hope I've shown, improvements in and prevalence of DNA sequencing change our identity in the contemporary moment and in prehistory, challenging us at both chronological ends. If it has any meaning, then the self must establish somehow a relationship with changes that took place over millennia. DNA sequencing works to construct imaginatively a non-negotiable relationship. Whilst textual material might lie or be lacking, the DNA test in the popular imagination is basic proof. It suggests a surety of imagination. It challenges the archive, positing the validity of what is to be found in the body over what is to be found or not in the text. And so therefore it challenges a particular model of state organization. Genetic genealogy undermines the idea of nation, of modernity, and even of history. It enables possibly the membership of a new order, a new self born from scientific investigation of that which was inside all along. I'm not necessarily saying I subscribe to this, but these are the kind of implications, I think, for some of this. DNA testing seemingly writes back into history those who had been forgotten due to lack of textual evidence. It reminds us that Anzic I was a human being who lived and died, that Kenwick man existed in time and in relation to us. And DNA sequencing introduces a new epistemological understanding of the modern self's relationship to the past, not governed by the evidence of the state, but instead by sequence of genes. Selves can be imagined possibly in new ways, and the relationship to the new past is reordered. And I'm arguing, I guess, that DNA sequencing has warped our relationship to the past irrevocably. Popular use of DNA testing for ethnicity might suggest that DNA has an identity outside of history that provides a resistance to modern identity definitions. It renders a fundamental humanness back to the user. Yet DNA sequencing of ancient samples shows that we're still not really sure what this humanness even is. Thank you very much for listening.
The microphone?